In the previous couple videos, we've seen how the process of glycolysis fits into the picture of aerobic and anaerobic respiration. In this video, we're going to look at the overall process of glycolysis, look at each of the intermediates along the way, and think about the reactions that are happening. In the next two videos, we'll focus in on examining a few of these reactions in a little bit more detail, and then we'll end this section by thinking about the energy landscape of glycolysis, which plays a really important role in the points during glycolysis where we can have branches that lead to energy storage and lead to stored energy coming into the process of glycolysis. Okay, so what we're looking at here is the big picture of glycolysis. We're looking at the structure of all of the intermediates, as well as all of the important molecules like ATP, ADP, NADH, and NAD+, that play a role in the overall energy production process of glycolysis. Okay, what we see here is we're starting with glucose, and we're ending with the molecule pyruvate. And as we saw in the previous couple of videos, we know that's what happens in glycolysis. Glucose is converted to pyruvate, making a total of 2 ATP molecules and 2 NADH molecules. All right, so the very first step in this process is the conversion of glucose, our energy substrate, into glucose 6-phosphate. This happens by transferring a phosphate group from ATP and putting that phosphate onto the 6th carbon of glucose. So we make glucose 6-phosphate. We put a phosphate group onto carbon number 6 of glucose. This process converts one molecule of ATP into ADP. All right, so we now have our first intermediate in glycolysis. The second step is converting glucose 6-phosphate into fructose 6-phosphate. Okay, so you may recall that glucose and fructose are very much related. Glucose is an aldose, whereas fructose is a ketose. But otherwise, the orientation of the alcohol groups are the same. So this ends up being an isomerization of the aldose, glucose 6-phosphate, to the ketose, fructose 6-phosphate. In the next step, we now convert fructose 6-phosphate into a molecule that now has 2 phosphates, and this is fructose 1,6-bisphosphate. Okay? By the source of this phosphate is ATP, so in this step we're converting another molecule of ATP into ADP. So these first 3 steps, converting glucose to fructose 1,6-bisphosphate. These are called the investment steps, or the preparatory phase. We call this the investment or preparatory steps because we're consuming ATP, but we're doing so in a way that prepares the intermediates to be able to make energy, to be able to create ATP. Okay, so this is the end of our preparatory phase. The next steps are all leading off to a payoff of ATP. Okay, so the next steps are called our payoff steps. The first part of that is taking our 6-carbon chain of fructose 1,6-bisphosphate and converting that into 2 equivalents of 3-carbon chains. When this happens, we make 2 molecules, one called dihydroxyacetone phosphate, and the other one is glyceraldehyde 3-phosphate. Okay, so in this process, in this splitting process, we end up with 2 products, but ultimately we want to convert the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate because that's what we need to then move on in glycolysis. So there's actually an equilibrium here between the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate. These 2 molecules can very quickly interconvert using an enzyme that works very, very quickly. And we'll look at this enzyme in a later video. Okay? Once we've now made 2 equivalents of our glyceraldehyde 3-phosphate, we can now convert that to our first high energy intermediate. So we're converting glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Okay, so we're putting an extra phosphate onto our 3-carbon chain, now on the first position and the third position. But instead of using ATP this time, we're actually using inorganic phosphate. Okay, and inorganic phosphate is any form of phosphate. So it can be phosphate, hydrogen phosphate, dihydrogen phosphate, or phosphoric acid. And we know that in the biological pH range, that's most likely hydrogen phosphate and dihydrogen phosphate, because those are the 2 species that are in the highest abundance at neutral pH. Okay, but this molecule 1,3-bisphosphoglycerate is the first high energy intermediate that we make in glycolysis. And the reason this is so important, we now have a bond that contains a lot of energy. Enough energy, in fact, that it can drive the conversion of ADP to ATP. So forming these high energy intermediates are what's needed to make ATP from ADP. And this is the first place in glycolysis that we're able to do that. And remember, since we have 2 equivalents of 3-carbon chains, we actually create 2 equivalents of ATP when we convert our 1,3-bisphosphoglycerate to the next intermediate, 3-phosphoglycerate. 3-phosphoglycerate is then subsequently converted to 2-phosphoglycerate. So all that's happening here is we're moving the phosphate group from the 3rd carbon to the 2nd carbon. And then once we've made 2-phosphoglycerate, we can now convert this molecule to phosphoenol pyruvate. And this is our last intermediate in glycolysis, and it's also the second high energy intermediate. So this molecule now has enough energy built into the bonds where it can convert ADP to ATP. And when that happens, the phosphoenol pyruvate is converted to pyruvate. So like we saw in the first high energy intermediate, we actually have 2 equivalents of this since it's only 3 carbons, which means we are creating 2 molecules of ATP in this step. So overall, in the preparation phase, we are converting 2 molecules of ATP to ADP. But in the payoff phase, we have 2 separate steps that convert ADP to ATP. And each of those 2 steps convert 2 equivalents of ADP to ATP because we have 2 3-carbon chains. So the preparation phase is working with 6 carbon units, glucose and fructose. But in the payoff phase, we've broken that 6-carbon chain down to 2 3-carbon chains, so every subsequent step creates 2 products. So ultimately, we make 2 equivalents of NADH and 2 equivalents of ATP. In the next video, we will look at a couple of these reactions in more detail and think about the chemistry of how it actually happens.